Howdy, folks. Thanks for checking into Mr. Ulrich's Land of Biology. I am Mr. Ulrich. In this little notescast, we're going to be taking a look at how we can follow traits from one generation to another, and how we can illustrate that using what are called pedigrees. Now, when I ask people what do they think of when you think of pedigree, of course they think of dog food, because it's a brand of dog food. But that's not really what we're talking about, although it does make sense that the pedigree brand is taking advantage of our association with pedigree being high bred or high born. And what that really means is if you're showing dogs for a particular breed, they have to have particular characteristics. And so if they're going to be purebred, that means that you know that the parents were of the same breed, had the same characteristics. And if their parents were purebred, then that means that you had to know what their parents were, and so on and so on. And you're establishing kind of a family tree of those particular characteristics. I do love the names that some purebreds end up getting. All bees are honey bear. It's certainly can be pretty entertaining. These pedigrees can go a few generations back to really, like I said, establish that family history of the traits that they're looking for. When it comes to a pedigree for analysis for the transmission of a trait, the tables start to look like this. Um, Now, you won't have to necessarily memorize that the males are the squares. I always say males are blockheads, and the females are the round ones. What we do have to remember is that the filled in individuals have the trait phenotypically, they show the trait, and the clear ones do not show the trait, whatever that trait is. And this makes it a handy way to show how a trait can skip generations. Here it is in generation 1, in individual 2, but then no individuals in generation 2 have show the trait. But then boop, it pops back up again in generation 2, right here in the direct lineage in individual 8. So how could that be possible? Well, this is just what Mendel showed, right, that a trait can skip generations. If this trait is recessive, right, then we know that this individual up here had to have, we'll use little b's, right, had to be homozygous recessive for this trait, right? And that means, of course, that all of their offspring had to be heterozygous because they are phenotypically dominant. They're showing the phenotypically dominant trait, right? But because this mom, she donated a recessive allele to each one of her offspring, right? So it disappeared. Now that's in order for this individual down here, who is also homozygous recessive because they're phenotypically showing this trait that's capable of skipping generations. That means that this individual here in generation 4 had to marry another heterozygote. That's what this line here means, of course, is married, because we're being very traditional. And their child then got a recessive allele from one parent and a recessive allele from another parent. And so these become, like I said, a good way to explain how a trait, recessive trait, like blue-eyedness, can skip from one generation and then pop up in the next generation. This part of the presentation covers the first section on the Pedigree Analysis Lab that you should get out now so that we can go over it together. This is going to make the most sense if you have the Pedigree Analysis Lab from class. Yours might say Human Inheritance and Pedigree Analysis, but it's the same lab. I just split it up. This is the part 2 of that lab. The first part that you're going to get to here in part 2 or of this lab here is just to kind of show you again how to give you a reference on how pedigrees are set up so that you can follow. There is a question here. Make sure you answer the question. It's pretty straightforward. But I want to be able to go over this first one here with us all together. So if you don't have the lab, pause and get it out and answer the question, and we'll wait. Now, what I like to do with these pedigrees is I like to go through and do my knowns first. Now, this is an albinism pedigree, and just kind of as an aside, as kind of an artifact of the example that we're doing, albinism, I would think for the trait to show it in a diagram, since albinism is the lack of pigment, would be the clear one, right? The one that isn't filled in, and then normally pigmented would be the filled in, the black squares and circles. But being filled in means that they show the trait. And since albinism is a recessive trait, that means that generation 2, individual 6, since they are phenotypically recessive, showing they are an albino, they must be homozygous recessive. And I like to draw right on the pedigree itself. Now, again, using logic, if this individual is homozygous recessive, has 2 recessive alleles, they must have gotten 1 from one parent and 1 from the other parent. But both of these parents are typically pigmented. And that means that they must have at least 1 dominant allele. Now, we can go through, like I said, and put in all of our knowns, since all of these individuals are phenotypically typical. They have typical coloration. We can go through and give each one of them a dominant allele, right? 1 capital A. And then really it's just a matter of which one of these have recessive alleles. Now we know that the daughter here in the 3rd generation, number 2, right, she is also homozygous recessive. So she can't get both from one parent, couldn't get both from mom. She's going to get 1 from mom, one of the 2 here. But that also means the dad had to have 1 to donate as well. Now, that's pretty much it for this one, because we can't tell what the second allele is going to be. From this mate, from this couple, if you do the Punnett square real quick, you see that all of the combinations are possible. I mean, there's one more here. There you go, finish it. Right, that all of the different combination, homozygous dominant, homozygous recessive, heterozygous. So we can't tell on any of these. Now you can go through and fill this out on this side. All right. The PTC taster diagram is a little bit different, so you got to read the instructions. I'm going to let you finish that on your own. All right, that's enough. I think you should be able to, like I said, finish those part 2 questions and finish the PTC pedigree analysis. If you have questions, feel free to email me, drop them in the comment section. Emailing me would certainly get a more direct response. Other than that, I'll see you in class.